Assalamu alaikum, good morning everyone. In today's session I'm going to cover the adaptive immune response. The objectives of today's session are to define the adaptive immune response, to describe the major components of the adaptive immunity and their functions. We have cells and proteins that are key players in the cell-mediated and humoral immunity respectively. Then to describe how the cells of the adaptive immune system recognize antigens and discriminate self from non-self. And then to illustrate the network between innate and adaptive immune responses. And finally, to briefly describe the consequences of aging on the immune system or the immune response. To remind you, immunity is the ability of the human body to resist almost all types of organisms or toxins that tend to damage tissues and organs. The immune defenses, as you remember, or the immune defenses of the body are classically subdivided into non-specific, innate, and specific adaptive or acquired immune responses. We covered the innate or the non-specific immune response. Today we'll start to talk about the adaptive or acquired immune response. Substances that are recognized as foreign and provoke an immune response are called antigens. Here, as you can see, we have a microbe that has antigens on its site. And to these antigens, the antibodies that are produced by the immune response can attach. The adaptive immunity describes the ability of the body to adapt defenses against the antigens of specific bacteria, viruses, foreign tissues, toxins, and etc. So the body will develop or adapt a defense mechanism against those specific antigens of bacteria, viruses, foreign tissues, toxins, and so on. The adaptive immunity has two properties that distinguish it from the innate immune response. These are specificity for foreign molecules which act as antigens. So the adaptive immune response reacts to certain specific antigens. This involves distinguishing self molecules, normal, that are not antigenic, from non-self molecules. So the adaptive immune response will not attack or will not discover and attack our own antigens. It will only discover the non-self or the foreign molecules or the foreign antigens. Another feature that distinguishes the adaptive immune response from the innate immune response is the memory. The adaptive immune response develops memory for previously encountered antigens. So on the next encounter, the immune response will be faster and more efficient in eliminating that antigen. The antibodies of the immune response or the adaptive immune response, as you can see here, can develop to the cell wall polysaccharides, to the pilus proteins, to the cell wall, to the flagellum, and so on. Not all foreign substances are antigenic. For example, our body will not develop antibodies against glass. Molecules or parts of molecules tend to be antigenic if they are foreign, not part of our bodies, organic molecules that are structurally complex. Proteins are usually complex and form many of the most potent antigens, and large. So the higher the molecular weight, then it will be more antigenic. Antigens can have multiple antigenic determinants called epitopes. For example, here this is an antigen that has multiple epitopes. Each epitope on its own is capable of producing an immune response, and entire microbe may act as an antigen, but typically just certain small parts or epitopes of the large antigen complex trigger an immune response. So we don't need the entire microbe, just part or small part of an antigen that is called epitope can trigger and induce an immune response. The adaptive immune response cannot get started without the aid of non-specific phagocytosis that occurs in the innate immune response. So the adaptive immune response needs the phagocytosis, which is part of the innate immune response. How? The phagocytic cells that initiate the process are called antigen-presenting cells. So we need the phagocytes of the innate immune response to present the antigen to the cells of the adaptive immune response. So here, as you see, bacterium with the epitopes or antigens will be engulfed by phagocytes, and then these phagocytes will present the bacterial antigen to the cells of the adaptive immune response, and we call these cells that present the antigen to the cells of the adaptive immune cells, the antigen presenting cells, the APCs. As you can see here, after phagocytosis, the antigen presenting cells will bring the antigen to their surface and present it to the cells of the adaptive immune response. Antigen presenting cells, APCs, are mostly dendritic cells and macrophages, and these antigen presenting cells link the innate immune system and the adaptive immune system. The dendritic cells are usually found in tissues in contact with the external environment, and they are the most potent of the antigen presenting cell types. Here you can see dendritic cells that has branched projections called dendrites. That's why we call them the dendritic cells. These projections are similar to those present in the neuron, but they have nothing to do with the neurons. The dendritic cells are not specialized neurons, so be careful. As an antigen presenting cell engulfs and destroys a foreign invader or pathogen, it isolates the antigens those cells display. So it will digest the cell and take away the antigens to present them on their surface. The antigen presenting cells then present the foreign antigens to a specific T lymphocyte called the helper T cell. It is also known as CD4 cells. So after the digestion of the microbe or the foreign invader inside the antigen presenting cells, the antigens of those microbes will be presented on the surface of the antigen presenting cells, and these antigens are presented to special types of lymphocytes called the helper T cells. As I said, they are also known as the CD4 cells. Once stimulated, here we can see that the antigen presenting cell is presenting this antigen here to the inactive helper T cells. So once the antigen is presented to the T cells or the helper T cells become active. Activated helper T cells are capable of activating other lymphocytes to become cytotoxic T cells. We also call them the CD8 cells, which directly kill the foreign invader. So the cytotoxic T cells can directly kill the foreign invader, and also they can stimulate other lymphocytes, or the activated helper T cells can stimulate other lymphocytes to become B cells, which make antibodies that kill or helps killing foreign invaders. The activated B cells and activated T cells form the two arms of the adaptive immune response, and these two arms are the antibody-mediated immunity and cell-mediated immunity. So adaptive immunity has two arms or two parts. These are the antibody-mediated immunity and the cell-mediated immunity. Helper T cells aid in both types, and both types work together to form the specific bodily defenses or the adaptive immunity. As you can see here, we have an antigen or infection with the antigen, or any invader that has antigens. This will stimulate the innate or non-specific immune response that we covered in the previous lecture, and also the specific immunity and the immune system. As the initial contact with the antigen, the macrophages and the dendritic cells will migrate to the site. They will engulf the invader or the pathogen and present its antigen to the surface, where the helper T cells will recognize that, or the helper T cells become activated, and then the helper T cells in turn will activate the cell-mediated immune response and the antibody-mediated immune response. The cell-mediated immune response is mediated by, the antibody-mediated immune response is mediated by B cells that produce antibodies, while the cell-mediated immune response is mainly mediated by other T or other types of T cells. Cell-mediated immunity involves the production of cytotoxic T cells that directly attack invading pathogens, foreign invaders with the antigen that is harmful to us, particularly intracellular pathogens like viruses and some cancer cells. So the cell-mediated immunity is mediated by cytotoxic T cells and directed against virus-infected cells and some cancer cells. In the cell-mediated immunity, suppressor T cells and memory T cells are also produced. The other arm of adaptive immunity is the antibody-mediated immunity that involves the production of B cells that transform into antibody-making plasma cells. So once activated, B cells will transform into plasma cells, and the plasma cells will produce and release different types of antibodies. Antibodies circulate in the extracellular fluid, and also during the activation of the B cells, B memory cells are also produced for the next invasion or the next encounter with the same antigen. Now let's talk in details about cell-mediated immunity, and then later on about the antibody-mediated immunity. In cell-mediated immunities, the T cells or T lymphocytes play a major role. So roles of T lymphocytes in cell-mediated immunity: we have the helper T cells. They secrete cytokines that enhance the activity of B cells and other T cells. Helper T cells enhance the activity of macrophages and natural killer cells of the innate immune response. Also, another type of T lymphocytes are the cytotoxic T cells. They kill virus-infected cells, abnormal cells, and bacteria. The suppressor T cells. Another type of T cells, they secrete cytokines that suppress the activity of B cells and other T cells. Our immune system has the remarkable ability and responsibility of responding appropriately to a wide variety of potential pathogens in our environment. The proteins that are used as cell markers to flag self from non-self are called MHC molecules and are coded for by a group of genes called the major histocompatibility complex. T cells have antigen receptors that recognize antigens only when it is associated with major histocompatibility complex. So the T cells cannot recognize the antigens that are presented on the surface of antigen-presenting cells unless they are associated with the major histocompatibility molecules, as we'll see in the coming slides. The major histocompatibility molecules bind antigens within the cell and transport antigens to the surface where it can be recognized by the T cells. Major histocompatibility genes are diverse and vary greatly from individual to another individual. There are two general classes of MHC molecules, and at least one or the other or both are found on the surface of all nucleated cells in the body. Class 1 molecules, MHC1, are built into almost all body cells and are used to present non-self proteins from bacteria, viruses, for example, to cytotoxic T cells. So the class 1 molecules are present in all body cells and they are responsible for presenting the non-self proteins that are part of bacteria or viruses to the cytotoxic T cells. The class 2 molecules or MHC2 are only found on antigen presenting cells, surfaces of macrophages, activated B cells, activated T cells, and the thymus cells. So MHC2 molecules are only present on the surface of the antigen presenting cells like macrophages and activated B cells and T cells, as well as the thymus cells. Both classes are important for antigen processing and presentation. Final maturation of T cells depend on the contact with the MHC molecules. So when they bind with type 1 MHC, cytotoxic T cells are activated or become mature. When they bind with type 2 MHC, helper T cells or CD4 cells are activated or become mature. Now the role of MHC molecules in antigen presentation, as I explained earlier. This is, for example, here we have a virus infected cell, any cell in the body. We said that class 1 MHC is expressed or is present in all nucleated cells of the body. And let's say we have one cell in the body that was infected with the virus. The newly produced virus protein will be divided or fragmented and then associated with class 1 MHC and presented to the surface of the virus infected cell. Now the cytotoxic T cell that has a T cell receptor specific for the virus antigen fragment, with the help of CD8 will recognize these virus infected cell. The cytotoxic T cell become active and will destroy the virus infected cell. Here the other example on the right side of the slide, a macrophage or dendritic cell, which is an antigen presenting cell that has the class 2 MHC. So if we have a virus or bacterium that was ingested during phagocytosis. The antigen was extracted, fragmented, then bound to the class II MHC and presented to the surface of the antigen presenting cell. Then the helper T cell that has the specific T cell receptor for that virus or bacterium antigen, and with the help of CD4 will recognize that viral antigen that is bound to the class II MHC, and the helper T cell now becomes activated. So MHC molecules have binding sites for antigens, as we saw in the previous slide. MHC plus antigen will then translocate to the surface, or will present the antigen to the surface. Type 1 or class 1 MHC binds antigen in infected cells or tumor cells and present it to the surface. And the infected cell or tumor cell can be any nucleated cell in the body. So it shouldn't be, or it is not necessarily one of the immune response cells, any cell, an epithelial cell, a neuron, a muscle cell, or whatever. Type 2 or class 2 MHC binds or molecules bind antigens taken into cells by phagocytosis or receptor-mediated endocytosis, which can occur only in cells that perform these functions, like B cells, T cells, and macrophages. Class 1 MHC molecules on the infected cells or tumor cells present the fragments to the cytotoxic T cells, which directly kill those cells. Type 2 MHC molecules on the antigen presenting cells present the fragments to helper T cells, which in turn stimulate an immune reaction from other cells. So the class 1 with the antigen, or will present the antigen to the cytotoxic T cells that will directly kill the cell that is presenting that antigen, either virus or tumor. And class 2 is a molecule that presents the antigen on the surface of antigen presenting cells, and this will stimulate helper T cells. Here we can see in this example the activation of cytotoxic T cell. The infected body cells present the antigen using the MHC1 molecule. So the cell is infected, for example, with a virus. The antigen will be processed, bound to the MHC1 molecule, and presented to the surface of the cell. This will be recognized by the cytotoxic T cell that has a T cell receptor specific to that antigen that will bind the virus infected cell and will activate the cytotoxic T cell to release granulolysin, perforin, that will put channels inside the cell membrane of the infected cell and cause the lysis of the virus infected cell. Helper T cells also coordinate the immune response. The events of activating the helper T cells is when the antigen presenting cells like macrophages or B cells present class 2 MHC foreign antigen complex on their surface. This complex will be recognized by helper T cells that have the receptor specific for that foreign antigen. Now the activated, after binding, the macrophages and B cells will secrete interleukin-1, which induces the proliferation and differentiation of helper T cells. Helper T cells differentiate into secretory cells. We call them also the effector cells. They release cytokines that stimulate and regulate the immune response, and also they differentiate into memory cells. memory cells. So the next cell-mediated immune response will be when the same antigen is encountered, will be faster and more efficient. Cytokines. We mentioned the word cytokines in the previous slide. So what are the cytokines? Cytokines are chemical signals or chemical molecules that are released from one cell and influence another cell. They are small protein hormones that control cell growth and differentiation. Examples of cytokines that you will encounter in different courses in phase 2 inshallah are interferon, interleukins, erythropoietin, and the tumor necrosis factor. Here you can see in this table examples of cytokines secreted by activated helper T cells like interleukin 2, interleukin 4, interleukin 5, interleukin 10, and interferon gamma. They have effect on different target cells with different responses of those target cells to those cytokines. You don't have to memorize. I'm just giving an example of the cytokines and their target cells and what is the effect of those cytokines on the target cells. There is something called clonal selection. Clonal selection is the process by which a lymphocyte proliferates and differentiates in response to a specific antigen. So the lymphocytes that proliferate in response to a specific antigen are different from the lymphocytes that proliferate and differentiate to another specific antigen. So the first one is the clone of lymphocytes that is specific to that first antigen, while the second group of lymphocytes is another clone that is specific for the second antigen. A clone is a population of identical cells all recognizing the same antigen as the original cell. Lymphocytes undergo clonal selection to produce effector cells, the active helper T cells, active cytotoxic T cells, and plasma cells that die after the immune response. Memory cells that do not participate in the initial immune response but are able to respond to subsequent exposure by proliferating and differentiating into more effector and memory cells. So the memory cells are not for the first encounter but for the subsequent encounters. They just carry the memory of the immunity or the immunity to the first encounter with the antigen for next attacks. We finished the cell-mediated immunity, and let's talk about the antibody-mediated immunity. The antibody-mediated immunity, or the key player in antibody-mediated immunity, are the B cells. B cells can be activated by direct recognition of antigen through B cells receptors or through the helper T cell activation when they are activated by antigen presenting cells that present an antigen on their surface. So here we can see inactive B cell with B cell receptors on the surface. They can be activated by direct recognition of the antigen on the surface of the microbe or through the co-stimulation of the helper T cell using several interleukins. So the activated B cells will form a clone of B cells. The clone of B cells is formed of plasma cells that produce and secrete the antibodies and memory cells for the next or subsequent encounter with the same microbe. So for the next infection with the same microbe. Activated B cells undergo clonal selection to become antibody producing plasma cells that will produce antibodies specific to the antigens present on the surface of the microbe. Antibodies are also called immunoglobulins, are produced by plasma cells through antibody-mediated immunity. Antibodies are composed of 4 peptide chains, 2 heavy chains and 2 light chains. You can see here 2 heavy chains, the yellow and the blue one, and the 2 light chains that are in red, and each antibody or disulfide bonds link the chains together in a Y-shaped arrangement. There is something called the variable region or the antigen binding region that gives the antibody its specificity. Here we can find the antigen binding sites, and these are the variable regions, and they are different from antibody to another antibody. The stem here, the stem region is similar for each class of antibodies. We have 5 classes of antibodies, and the stem is the same in each class of the antibodies. Some of the ways antibodies are effective in mediating the immune response include neutralizing a bacterial or viral antibody or a toxin by covering the binding sites and causing agglutination and precipitation, so making what was soluble insoluble. So they neutralize the bacterium or the virus or the toxin to decrease its aggressive behavior. Another way antibodies function is opsonization. So they attach to the surface of the microbe, so just like they coat it, and when the phagocyte encounters the microbe, it will recognize the microbe that is tagged with the antibodies more easily. Complement system activation, by activating the classical pathway of the complement system that has several functions, and I will mention those functions in one of the coming slides. So the antibodies protect against diseases by neutralization and agglutination. Here, neutralization of the toxin or agglutination of the bacteria. Here, the opsonization, they tag the microbe so the phagocytes can easily spot the microbe and engulf it, or by activating the complement system that will insert pores or holes in the wall of the microbe, and this will cause lysis of the microbe. The complement system is a series of blood proteins that often work in conjunction with antibodies. The system can be activated by multiple pathways in a stepwise or cascading fashion. It encourages vasodilation and inflammation, antigen opsonization, and antigen destruction. The main proteins in the complement system are C1 to C9. You can see here that the complement, we saw that antibodies can opsonize bacteria or microbes. Also the complements, especially C3b complement, can opsonize the microbes that will be recognized by the C3b receptors. So one of the functions of the complements is opsonization, which is similar to the effect of some antibodies. Another way is inserting holes in the wall of the microbe, causing its lysis. These holes that are inserted with the help of the complement system are called membrane attack complexes. They form as a result of activation of the complement cascade, and these membrane attack complexes are then inserted in the wall of the microbe, causing cytolysis or lysis of the microbe. As I said, the membrane attack complex results in lysis of the cell. There are different classes of antibodies. We have IgG, we have the IgA, we have IgD, IgE, and the IgM. If you notice, the IgM is a pentamer. It is composed of 5 units of antibodies. There are 5 classes of antibodies. IgG is a monomer with 2 antigen binding sites. It comprises 80% of the total antibodies that are present in our body. The only class that is able to cross the placenta. So maternal antibodies of the IgG class can cross the placenta and give innate immunity to the fetus. The IgG antibodies provide long-term immunity. The IgM is a pentamer, as we saw in the previous slide, with 10 antigen binding sites. A monomer has 2 sites or 2 antigen binding sites, so pentamer has 10 antigen binding sites. It is a great activator of the complement system, but has a short-lived response. It is the first antibody to appear in an immune response. So the first antibody that will appear in response, for example, to bacterial infection is the IgM class. IgG is a dimer with 4 antigen binding sites. It is prevalent in body secretions like sweat, tears, saliva, breast milk, and gastrointestinal fluid. So it is present in body secretions. The IgE is a monomer involved in allergic reactions, comprises less than 0.1% of total antibody in the blood. And finally, the IgD is a monomer with a wide array of functions, some of which have been a puzzle since its discovery. Please remember that IgG, IgE, and IgD are monomers. They each has 2 antigen binding sites. IgM is a pentamer that has 10 antigen binding sites. IgA is a dimer and has 4 antigen binding sites. The antigen that is important for the allergic reactions is IgE. The antigen that is the antibody that is present in the body secretions is the IgA, and the first antibody that appears in response to infection or as part of the immune response is the IgM, but it has a short lifespan. Thousands of memory cells exist after initial encounter with an antigen, and this is called immunological memory. With the next appearance of the same antigen, memory cells can proliferate and differentiate within hours. This graphic shows that serum antibody titers are much higher and much faster on the second response, and this is the advantage of the memory or the immunological memory. As you can see here in this graph, first of all, the red line represents the level of the IgM over time, while the blue line represents the level of IgG over time. If you can see, as I said in the previous slide, the first antibody that appears in response or in the immune response is the IgM, then the IgG. The IgM will go down because it has short lifespan. The IgG will not go back to the baseline because it has a longer lifespan. On the second exposure to the same antigen, due to the presence of immunological memory, the IgG will rise immediately, several folds higher than in response to the first exposure. The response of the IgM will be the same as the first exposure, and as we said, it is short-lived, so it will go down to the baseline shortly, but the IgG level will increase several times and will remain high for longer time. Here we can see a summary of the adaptive immune response. As you can see, the primary lymphatic organs, the red bone marrow give us the mature B cells and the pre-T cells that will reside into the thymus, where they will undergo more maturity, and then they will be released to the secondary lymphatic organs and tissues. Upon exposure to an antigen, the helper T cells will become activated, and also the cytotoxic T cells, the B cells, and this will lead to the activation of, according to the type of the antigen and the infected cell or the affected cell, into cell-mediated or antibody-mediated immune response. Don't forget that memory cytotoxic T cells and memory B cells will develop in each case. Gaining immunocompetence or becoming immune within the framework of innate and adaptive immunity we have discussed. There are a number of designations for the ways we can become immunocompetent. We can be immunocompetent by natural immunity, which is not gained through the tools of modern medicine like vaccine, which is artificial immunity. So there is natural immunity and artificial immunity, and I will give examples about them in the next slide. We have also active immunity, which refers to the body's response to make antibody after exposure to a pathogen or an antigen, and there is something called passive immunity. The body simply receives antibodies that have been preformed outside the body. Active immunity is long-term or long-lasting. Passive immunity is short-term. It will be lost once the given antibody's lifespan ends. Examples of gaining immunocompetence: we have natural active immunity, contracting, for example, hepatitis A, getting hepatitis A through food or water, and the production of anti-hepatitis A antibodies. So this is natural because we got the infection naturally by consuming contaminated water or food with hepatitis A. And active because the ingested hepatitis A virus will stimulate the immune response to produce anti-hepatitis A antibodies, so it is an example of natural active immunity. Natural passive, we said that IgG or antibodies of the IgG class can pass through the placenta. So a baby receives antibodies from his mother or her mother through the placenta, and this is IgG, and breast milk, which is IgA. We said the IgA is present in body secretions, including breast milk. Artificial active immunity: a person receives a vaccine of an attenuated, changed or weakened pathogens, like the BCG, the vaccine for tuberculosis, and this will stimulate the body to form an antibody. So the person didn't contract an infection, but he received the weak antigen by an injection. An example of artificial passive is just the injection of pre-prepared antibodies obtained from another person and then injected into the blood of the recipient to give him an artificial passive immunity. As I said, natural immunity is long-lasting. Active immunity is long-lasting compared to the passive immunity. So this, what is the effect of aging on the immune system? With aging, atrophy of the thymus gland results in decreased T helper cell populations and the diminished mediation of the specific immune response. There is a resulting decreased B cell response and decreased number of T cytotoxic T cells. Compromised immune function with age results in increased titers of autoantibodies and increased incidence of cancer. Both contribute to overall mortality rate in old people. With this slide, I end the lectures of the lymphatic system and immunity. I hope you got most of the concepts. I will appreciate receiving or sending your questions by email. Thank you very much for your patience, and goodbye.